All right, everybody. We're going to wrap up chapter 4 in preparation for you taking your first exam next Tuesday. So, chapter 4, where I left off, we were talking about the Hardy-Weinberg equilibrium. And if you recall from our lecture on Tuesday, the Hardy-Weinberg equilibrium establishes kind of a null hypothesis or a null prediction for what our genotype frequencies in a population are going to be if we are given or we have the allele frequencies. And so in this case, we have two different alleles, A1 and A2, and you'll see that in graph B, the frequency of those gametes is 30% and 70% respectively. If you will remember from your intro bio course, you learned how these allele frequencies can be used to predict genotype frequencies. Okay. Remember, every individual like you that is a diploid has two copies of every gene. Those two copies might be the same allele, for example, A1 and A1 that you got from each parent, or they might be different. You might have A1 and A2. You might be a heterozygote. The Hardy-Weinberg equilibrium uses the frequency of the alleles in the population to make an overall prediction of what the frequencies of the genotypes will be in that same population. And if you just think about what you see in the bottom right in C, these allele frequencies can be used using the product rule of probability to calculate expected genotype frequencies. Because in this instance, there are only three possible genotypes, and there are two alleles that create all three of those possibilities. We use their frequencies to determine if a random male and female from this population get together and have one offspring, what's the probability that that offspring gets an A1 from one parent and an A1 from the other parent? Well, if the frequency of those A1 alleles in the population is 30%, then there's a 30% chance that that parent gives each parent gives an A1 to their offspring. And so we multiply those two frequencies, 0.3 times 0.3, to get a 9% probability of the offspring of that breeding having the A1 homozygous genotype. If that's true of all individuals in the population, which the Hardy-Weinberg equilibrium equation suggests, then that product rule of probability can be used not only to calculate the probability that a single individual is homozygous for A1, but that's also predictive of the overall frequency of A1 homozygotes in the population. So not only does every individual have a 9% chance of being homozygous for A1, if you look at all the offspring for the whole generation, for this generation, 9% of them you would expect would be homozygous for A1, right? If every individual has a 9% chance, then you expect 9% of them to be homozygous for A1. Just think about it. If there are 100 offspring, every one of them has a 9% chance. How many of them will be homozygous for A1? Probably 9 out of 100, or 9%. Okay, so you can use everything that's on this slide illustrates how you can calculate genotype frequencies from allele frequencies using the Hardy-Weinberg equilibrium equation. Okay, I'm not here to teach you this equation again. You've seen it before. It's explained here. There are plenty of short videos you could watch on YouTube, or there's an example in your text that you could use to make sure that you understand how to calculate the expected frequency of a genotype given allele frequencies. Okay, I won't use examples with more than two alleles. You can do that in genetics. So nothing more complex than what you've seen here, but you should understand the Hardy-Weinberg equation. Now, the reason we're talking about the Hardy-Weinberg equilibrium equation is because, as I told you, this is kind of a null hypothesis, and the assumptions associated with this Hardy-Weinberg equation are that the population that follows the expectations of the Hardy-Weinberg equilibrium equation, it would have to be a very large population There would have to be no mutation in the population, no selection, completely random mating, and no migration. Now, why all of these things are requirements of the Hardy-Weinberg equilibrium equation for it to hold true would make sense if you think about it, okay? If there are only 2 individuals in the population, then those allele frequencies, you're going to have a lot more random chance playing into what the frequency of a genotype is in the next generation, and the allele frequency is not going to be as predictive, right? If those 2 parents have only 1 offspring, then even if there are 3 possible genotypes, some of those are going to occur at a frequency of 0, because there's only 1 offspring born, and not every genotype can be represented in that one. It assumes no mutations, right? Mutations will change allele frequencies. It assumes no selection. Selection, of course, would favor some genotypes over others, or some alleles over others. And that might mean that some alleles don't survive to reproduce and move on to the next generation, because they make the individuals less fit, less able to survive, less able to find mates. It assumes that mating is completely random. If that weren't the case, if males are being picky or females are being picky and choosing their mates based on phenotypic traits that are determined by their genotypes, then some genotypes, some alleles, have a better chance of going to the next generation than others. And that probability that they go to the next generation is not only associated with their frequency, right? The allele frequency doesn't predict genotype frequency because there's non-random mating. Some of those alleles, they might be very common, but they're not interesting to potential mates. It also assumes no migration. And so we were just talking about a population in which the allele frequencies were 30%, 70%. Well, what if a bunch of migrants from a mainland area fly out to or swim to an island where there's a population of a particular species that has these allele frequencies of 30% and 70%. But the mainland where they came, the frequency of the A1 allele is instead, let's say it's 0.9. It's much more common on the mainland. That influx of those, that disproportionately high frequency of the A1 allele to this island will dramatically change the expected frequency of genotypes in the next generation. So all of these things are going to throw off the Hardy-Weinberg equilibrium equation. It will no longer be accurately predicting genotype frequencies if any of these things happen. And all of these things are the things that we're going to be learning about. These are the mechanisms of evolution that drive change in populations over time. So if we think about these allelic frequencies and genotype frequencies changing over time in populations, one of the genetic things that I want you to be aware of is the linkage, genetic linkage, and the process whereby those linkages can be recombined. Okay? So when we are talking about genetic linkage and recombination, we're speaking of alleles for different genes that are associated with one another in genotypes and in phenotypes of individuals at a disproportionate rate. Okay? And so what I mean by that is, imagine this scenario here where we have genes A and B. And for each of these genes, there are 2 different alleles. So there's A1 and A2, B1 and B2. Okay? Now, so there are 2 different ways these could be passed to offspring. Given that the parents have either 1 and 1 or 2 and 2, when they produce offspring, those offspring might be able themselves to produce gametes that have A1 with B1 and A2 with B2, as you see on the left, or they might be able to create other combinations like A1 with B2 and A2 with B1, as you see on the right. Those are combinations that did not exist in the parents or the offspring that are producing this gamete. Okay. So there's a new combination of alleles. Those alleles are recombined in these gametes. Now, as you know, you have only 23 different pairs of chromosomes, 46 total chromosomes, but you have tens of thousands of genes in your genome. And so what you necessarily can deduce from that is that each of your chromosomes has many, many genes on it. And as you know, mutation can lead to different forms of genes being established within a species and a population. And so if there are different genes on a single chromosome, and each of those genes has multiple alleles, just like these two genes each have two different alleles, those alleles of different genes might be physically connected to one another. They might be linked to one another. And so this is what we call linkage, all right? So because, as I've said, not every gene has its own chromosome, these chromosomes have multiple genes, and when those genes have multiple alleles, you end up with linkage, genetic linkage, okay? So think about the previous slide. When your mother gives you a chromosome that has A1 on it, it might come with B1, okay? They're on the same chromosome, okay? So if we look back at this slide, this offspring in the middle, the offspring has A1, B1, and A2, B2. They have 2 copies of A, 2 copies of B, all right? If those A's and B's, if the gene A and gene B are on the exact same chromosome, then when this offspring in the middle produces gametes, it could only produce the gametes on the left, okay, unless something we call recombination happens, okay? So this physical linkage, when they're physically on the same chromosome and they are inherited together, you get A1 with B1, you get A2 with B2, okay, on the same chromosome. We can end up with what we call linkage disequilibrium. So here's an example that might gel with your understanding of the frequency of certain phenotypic traits within the human population. So I want you to think about hair color and eye color. And we frequently think about blue eye color being associated with blonde hair, right? So blue eyes are most frequently associated with blonde hair. But blonde hair, I actually had blonde hair when I was young. Blonde hair, when I had hair, is associated with brown eyes. Not uncommon, I have brown eyes. But black hair, individuals with black hair also have brown eyes. Those both occur at pretty high frequencies. So those first 3 combinations are quite common. Now this 4th combination of black hair with blue eyes, this is a genotypic combination that, a phenotypic combination that occurs at a very low frequency. And why might that be? Well, think about it. There's a gene, let's just pretend like there's a single gene that determines your hair color. And there are multiple alleles. There's blonde and black in this case. And then there's a gene that controls your eye color, brown or blue, okay? So we have 2 different alleles for hair color, 2 different alleles for eye color. But think about the origin of that allele that creates blue eyes. This is, it seems, a very recent mutation in the human species. And if that mutation that created this new allele occurred on a chromosome that was in an individual that had blonde hair, right? That mutation is on a chromosome where the hair color allele is blonde. Then when you get the blue-eyed allele, you're getting the blonde-haired allele on the same chromosome. And unless that linkage is broken up, you're going to see a disequilibrium, just like we have illustrated on this slide. The frequencies are not balanced across all 4 possibilities. That's this disequilibrium that we're talking about. The disequilibrium being due to linkage, and hence the term. So if we have 2 different chromosomes here, notice this is, let's say, your chromosome number 1 and your chromosome number 2. These illustrate different genes on different chromosomes. So the bands on these chromosomes are frequently illustrated to represent different genes that occur across the chromosomes. And you'll notice that these two different chromosomes have very different things on them, right? They're different genes, different stretches of bands that are of different sizes. So on chromosome 1, you might have your hair color and eye color genes, and there are lots of different alleles in the human population. On chromosome 2 on the right, there might be the genes for, let's say, whether or not you'll go bald, whether or not, let's say you have moist or dry earwax. These are things that are genetically controlled. So the genes that are on these chromosomes exhibit linkage, okay? And the way in which that linkage can be broken up is through a process of recombination, where two different copies of chromosome number 1 in mitosis or meiosis, and it's meiosis that we're generally interested in, they line up in meiosis 1, and those chromosomes get tangled up and cross over and exchange ends, okay? And when they do that, when chromosomes cross over, I think I might have an image of this here in a second. Yep. As you see down on the bottom in the middle, when these chromosomes swap ends, and these are sorts of swaps in which things get mixed up or lost. When these chromosomes interact and swap, then the blue-eyed allele, for example, might jump over to a chromosome that has the black hair allele, and vice versa. The blonde-haired chromosome gets a brown-eyed allele, whereas it used to have the blue-eyed allele. Okay, so when those chromosomes, I encourage you to Google and look up linkage disequilibrium and crossing over and genetic recombination. It's up at the top of this slide. We can create these new combinations of alleles through this crossing over. Now that crossing over is much more likely if those genes are really far apart. If they're super close together, it's not going to happen very often, right? The crossing over, that mix-up, that exchange would have to happen in a very small space. So you notice up on the top left, you see that R, recombination rate of 0.03. That means those two genes are really close to one another, and they very rarely, that's why that number is small, recombine. Very rarely do two chromosome ones come together and they cross over right at that spot in between the two. But look at chromosome 2 on the right. Those two genes that are really far away, that recombination rate is 0.4. That's pretty common, okay? That's a high frequency of recombination of chromosomes. And so that means if the genes are that far apart, the disequilibrium can be broken up pretty quickly. Okay, so I'm not going to ask you to draw this or identify it on an illustration. I just want you to understand that allele frequencies can change due to lots of things, all of those violations of the Hardy-Weinberg equilibrium equation. But there are some really interesting instances where those forces, let's say non-random mating or selection, might act on a particular gene, one of these that are illustrated on this slide, and that gene might, certain alleles of that gene might become more common, more frequent. But because of that linkage, they might bring with them alleles of other genes that weren't selected for directly, but indirectly, they go along and they increase in frequency too. Okay. So as the recombination rate increases, so that R number from the previous slide, the higher that R number is, okay, the further apart they are, the higher that number is, the faster linkage disequilibrium decreases. And you can see if things are really far apart on a chromosome, okay, then linkage disequilibrium can go from quite high levels of 0.25, and I won't get at why that's the max. They would, over just a couple generations, very quickly approach zero linkage disequilibrium, because those genes are on separate chromosomes or so far apart that they're effectively on different chromosomes and they're not linked at all. Now, If R is very low, meaning those genes are very close together on the same chromosome, you can see the difference between R of 0.1 and R of 0.01. If those recombination rates are really, really low, then it would take forever, as you can see, for linkage disequilibrium to decrease or even come close to zero. Okay, so mutations, we're not digging into all of the different types of mutations that happen. Some are at the bottom of this slide are illustrated in your text, and these are ones that you've learned about before. These are large chromosomal scale mutations where big chunks of the chromosome are lost or duplicated or flipped. Okay, there are large scale chromosomal mutations, and then you've of course already learned about small mutations where a G changes to a C. And as I showed you on Tuesday, that could lead to something quite dramatic, like the mutation that leads to sickle cell anemia. Okay. However, it's important to note that all of these mutations provide genetic variation within species. And if you didn't have that, if there's no genetic variation among any of us, okay, if we were all genetically identical, then there would be no differences between us that are heritable. And if that's true of all species, there are no heritable differences, then even if selection's acting on the population, let's say only the strongest or the fastest of us humans survive, okay? Just because, let's just pretend like I work out and run a lot, right? So I'm strong and fast and I survive and have offspring. It's only because I work out and run a lot. My kids might be lazy. Those traits that I have that were the result that were subject to selection, that's not heritable. And so my kids might be slow and weak and have low survival. Okay. We need genetic variation that causes the differences in the population. And those differences that are selected upon must be heritable. Okay. If you have variation in a population, but it's not heritable and there's selection, the population won't change. If you have no variation in the population and selection acts on it, the population's not going to change. You need variation, and you need that variation to be heritable. Okay. Now, when we think about mutations, we think about things like, I don't know, I think about, or you may think about, movies like the X-Men or those horrible Fantastic Four movies where those people went out into space and were exposed to all sorts of mutagens, and they mutated and they gained superpowers. Awesome. Well, you don't have superpowers, but you certainly have mutations. Okay. There are mutations that you have that neither of your parents had. They just made mistakes when they were making the gametes. Okay. So there are mutations in each of us, and many of them. Okay. All organisms mutate. Some of those mutations have beneficial effects. Some have no effects. Some have negative effects. What's interesting is that mutation rates are not the same across all species. So what you might think of as more primitive organisms like bacteria or archaeans have very low mutation rates. And things with large genomes like you and I, or invertebrates and plants, they actually have a higher mutation rate. Now, realize this is scaled by genome size, right? And the y-axis is rate per base pair, right? So this doesn't mean we have more mutations because our genome is larger. That mutation rate on the left is per base pair. Okay, so we have 10 times as many mutations on average than a plant would, the rate at which we accumulate mutations. Okay, so there are some interesting reasons or hypotheses as to why this might be, but we're not really certain. So what effects do mutations have? Well, when I talk about simple examples of different alleles, right? When I pretended like hair color and eye color are determined by one gene with two alleles, obviously that's not the case, right? We know that there are many more than just brown and blue eye colors, right? There are lots of other hazel and green and whatnot. The other thing that we need to note is that mutations to particular genes are generally pleiotropic. And that term means having multiple effects. Okay, so pleiotropic mutations have more than one effect. And an excellent example of the pleiotropic effects of mutations are the dwarfism that you see here in Peter Dinklage, so reprising his role in Game of Thrones as Tyrion Lannister, right? So as someone with achondroplasia, that is a mutation to a gene that's associated with growth factors, okay? And so that single mutation affects lots and lots of different things, okay? It affects aspects of, bone growth, of course, but a number of other physiological effects of that single mutation. So as you can imagine, this is a very clear example of a single mutation having multiple effects. Okay. Now, I mentioned earlier that mutations might have beneficial effects, no effect, or negative effects. Okay. Now, we know that mutations happen quite a bit, but what's interesting is that most mutations have, as you can see here, if you look at this graph long enough, you might read the caption in the text, most mutations have a negative effect. So the selection coefficient, or the x-axis here, describes how bad is this mutation for you, a particular mutation. How bad is it for the individual compared to what they were before the mutation? Okay, and so 0 on the far right is you're the same. There's no effect of this mutation. Anything to the left, that selection coefficient being negative means you are selected against because of that mutation. You have less fitness because of that mutation. And you can see that that's a wide range of, to the right, there are lots of mutations in this particular experiment that had a very small effect. It was negative, but it was very small, skewed to the right. But as you get farther and farther to the left, you can see that there are lots of mutations that have very negative consequences. All right. Now, if you look to the right of 0, things don't get that great. Okay. Most of these mutations that have an effect have no effect. Most of the mutations have no effect. Most of them that do have an effect, it's negative. And if they have a positive effect, it's a very small one, right? This graph doesn't go really far to the right side because there aren't single mutations to nucleotide sites that have enormous beneficial consequences, okay? Now, you can also look at this in the context of where did these mutations occur? Did they occur to stop codons? And you can see, what a stop codon is. If you have a mutation that creates a stop codon, that is a nonsense mutation, which you may have learned in the past. That's not a term you need to know for this class. But you should know what it means to create a stop codon in the middle of a coding sequence of a gene. And in doing that, you can see all those red ones way out to the heavy side of intense selection against that mutation. Non-synonymous versus synonymous mutations. You might remember from the codon chart that I showed you on Tuesday, and if you look at it a bit, you'll see that those amino acids, most of those amino acids in that chart, are coded for by multiple codons. Okay, there are multiple 3-letter nucleotide words that are used in coding sequences of messenger RNA. And if you change that third nucleotide position, a lot of times it doesn't change the amino acid that codon codes for. That would be a synonymous mutation. And you can see all those blue synonymous mutations. You don't change the amino acid. You don't, or you hardly change the polypeptide. And so there's no effect. So on average, synonymous mutations have 0 effect. Now, non-synonymous mutations, where the mutation, let's say, to the second codon position, you change that nucleotide, it will change the codon, it will change the amino acid, and those have a wide range of effects. Some of them have zero effect, some of them have extreme effects, and everything in between. So are mutations usually good or bad? Well, I think you can deduce from the previous slide and this one that the most mutations are going to either kill you or have no effect, right? If you randomly mutate something that's a critical portion of the genome, okay, then you are going to mess up something that is critically important. It's like the example I use is your car isn't, your car sounds a little funny. So you get up, open the hood, and reach in there and grab a wire and just yank it out, okay? What are the chances that you going in and randomly messing with some aspect of that complex machine is going to make it better? Very low, okay? What are the chances that it completely ruins the functioning of your engine? Far left of this slide, your fitness is 0 of your engine, pretty high, okay? Now, you might have just yanked out something that was, maybe it was a ground for your reverse light, and you're not even using that. You don't care if that works, okay? There's no change in the way your car runs. So, but you'll notice to the right of 1.0, there are very few mutations that lead to an increase in fitness. So most mutations have no effect or they're bad, all right? So it makes you wonder, how are we relying then in our exploration of evolutionary change on mutations to create beneficial traits that can be selected upon and the population changes? Well, this is why populations are enormous, okay? Or this is possible because populations are so large, because there are so many mutations within the population that many individuals die, many individuals have lower fitness, but it's those few that have slightly higher fitness and they have slightly more offspring, and their offspring have slightly higher fitness and slightly more offspring, that leads to this gradual change in the population over time. Where mutations happen does matter, and so we are primarily interested, really only interested in germline mutations. Mutations to the gametes that change the single copy of a gene that is going to be given to the zygote that is formed, such that the entire organism carries that mutation and will pass it on to the next generation. In this class, we are not interested in somatic mutations. I go out to the beach and I don't wear sunscreen and I get a mutation in my arm that gives me skin cancer on my arm. That area of my body is affected, okay? So it's not something that I will pass on to my offspring. It's only in that somatic tissue where the mutation occurred. Okay, the last principle I want to make clear to you is that evolution is not intentional, okay? Organisms don't strive to get better. They don't want to be more fit. That is not their desire, okay? Their fitness relative to other individuals in the population is a consequence of their genotype and their phenotype. And if it's high, if their fitness is high, they're going to have more offspring. It's not because they want it more, okay? And the mutations that underlie these phenotypic differences are really things that they have no control over, right? The mutations are completely random, right? And so just because let's say it would be beneficial for polar bears to be able to live in much warmer climates, because let's say it is getting warmer in their habitat around the North Pole, that doesn't, just because it would be beneficial for them to do that, doesn't mean that they will evolve that capability. It doesn't mean that mutations will arise within the polar bear population, making them capable of surviving extremely warm temperatures, okay? And so here's an example that illustrates one of two possibilities. The next one's in the next slide. But we have a plate of bacteria, So hopefully you guys will take a microbiology course while you're at Ole Miss if you haven't already. But if you have a bacterial plate there, and each of those blotches on the plate represents a colony of bacteria that is genetically distinct from all the others around it, and we can replicate that plate. It's actually pretty simple. You can effectively put a piece of paper or velvet on it, down on into that plate on the left, and then stick it on a clean replica plate in the middle there. And in 2 days or so, clones of those same colonies on the master plate will grow in the exact same places. Okay. So what we've done with the master plate here is make 3 copies of it. You can essentially photocopy colonies of bacteria doing this. All right. Now, if we add a virus to each of these 3 replica plates to see how this population handles this new pathogen, okay, what we expect is some individuals will survive and some individuals will not. So I want you to look at what happens in this slide. Look at the results in those 3 plates and what colonies survived. So here you've got 4 colonies in the top one, 3 in the middle, and then 5 in the bottom plate. So we have very different responses. That is not how this would happen, okay? Because remember, all of those colonies in the replica plates, each plate has an identical colony in the same place that is derived from an identical individual colony on the master plate. What we would see is on the master plate, if there were 4 colonies that before we even made the replica plates already possessed mutations that made them resistant to that virus, when you created the replica plates, those 4 colonies would be in the same place on each replica plate. You add the virus to all of the replica plates, and what you're going to get is 4 colonies in all 3 of those plates surviving. And it's the same damn 4 colonies, okay? Because that mutation already existed. And what I'm trying to get across here is that this population is not responding to this virus and rapidly evolving independently on each of these plates, rapidly trying to figure out who can survive, who can fight off this virus, and it be different like it was in this. That doesn't happen. So the mutations don't arise out of necessity. The mutations exist in the population. And when selection pressures come into the environment and act on that population, the population can only respond with the genetic variation that it already possesses. Okay. That is it.